Hello all. In this video, I will talk about a project that I've been developing for the last 3 years in the lab of my Gabrielle Zurich in the University of Lausanne, Switzerland, where I've been differentiating brain cells from human-induced pluripotent stem cells and investigating their applicability for neurotoxicity testing. This work is being developed under the IN3 project, where I've been working in the brain. The presentation is divided in two parts. The first, I will show the results that we have to try to understand if brain spheres are predicting the human response to chemicals, where I'm going to talk about Amiodarone and Part1 case studies. And then I will show you how we are trying to add microglia to our brain spheres. So for the first part, here you can see the timeline of my PhD. We are here now in the 4th year. And the first phase was to establish the model and characterizing it. Then we tested for the cytotoxicity of 5 out of 10 of the chemicals of IN3. Then we wanted to understand the dynamics of these chemicals in our system, and we collect a lot of samples for different tests, and we were also analyzing. We had the opportunity to send samples for TempoSeq. We are now in phase 4, where we are doing the data analysis of TempoSeq, and we are entering phase 5, where we are starting the paper preparation. In this slide, you can also see the secondments that I was able to do under the IN3 project in the University of Utrecht, L'Oréal, and Edelweiss Connect. So the phase 1 was to establish the model and to characterize it. We start from human-induced pluripotent stem cells. We differentiate them in neuroprogenitor cells, and then based on the protocol that David Pamiers published in 2017, we produce brain spheres. These brain spheres stay in culture for 8 weeks, and we can see a decrease in proliferation and stem cell markers and an increase in neurons and glial cell markers. So after establishing and characterizing the model, we started to expose this model to the chemicals from IN3. And we went for a repeated exposure of 1 week that started after 6 weeks of development, because it's when we can see the markers for neurons, astrocytes, and oligodendrocytes being present. And this week of exposure was followed by a week of washout, where there was no more addition of the chemical to the system. Here you can see the cytotox curves of amiodarone, doxorubicin, paraquat, valproic acid, and lead. To understand the dynamics of these chemicals in our model, we did different things, such as the TempoSeq, where we were able to send samples to understand these 5 compounds that I just told you about. We were also able to send samples to understand more the developmental time course of the brain spheres, and also to characterize which pathways can be activated in this model by specific activators in. Then we also perform qPCR and immunocytochemistry, and we send samples for distribution kinetics analysis. And then the phase 4 is data analysis. That's where we are. So for amiodarone, that is one of the chemicals of the IN3 list. It's the most commonly prescribed class 3 antiarrhythmic drug. Just in 2016, and just in the US, it was prescribed to more than 2 million people. It's known by blocking potassium, sodium, and calcium channels, and it has been in the World Health Organization World List of Essential Medicines for more than 20 years. The patients that take amiodarone can take it for a long time, and they report some side effects, such as tremor and ataxia, that we know that are linked to the central nervous system. So we thought that it would be interesting to try to understand the distribution kinetics of amiodarone in our model, and the neurotoxicity that could be produced by amiodarone in our model, to maybe understand a bit better what it can produce in the brain of a human. So the distribution kinetics, we were trying to understand the link in between the external dose that we add to our system and the amount that we actually can find inside the cells, amount of amiodarone. To do so, we collected samples of the medium before exposure, the medium after the exposure, the cells that were exposed to amiodarone, and also the plastic fraction, since we know that amiodarone can bind a lot to plastic. For the time of collection, it was 0, 1, 3, 6, 24, and 48 hours after the first exposure. Then we exposed the system 2 more times along the first 1 week of exposure, whereafter we collected some samples. And then we did 1 more week of washout period where the amiodarone was not added to the system anymore, and we collected samples again. When we when we try to detect amiodarone in our samples, what we can see is that in the first 48 hours, we see a decreased amount of amiodarone available in the medium. In the plastic, we can already detect amiodarone after 1 hour of exposure, and this amount keeps quite stable along all the exposure. And in the cells, we see that there's an increased amount of amiodarone. After the full week of exposure, we see a higher amount of amiodarone inside the cells, indicating an accumulation. And even when we remove the amiodarone from our system for 1 more week, we can still detect amiodarone inside the cells. When we do the mass balance of the medium cells and plastic fraction amount of amiodarone, we see that there's not a big variance along the first 48 hours, indicating that there's no production of metabolite. To be sure, we try to detect the most common metabolite of amiodarone and DEA, and we couldn't detect it in our samples. So now that we know that amiodarone is available to interact with the targets and inside the cells, we want to understand what could be the effect of an amiodarone exposure in the brain and in our model. We started by checking for cell type specific gene expression. So for neurons, we tested different markers, and we can see that in the general markers, there's a strong decrease in its expression after 1 week of exposure, mostly for markers regarding GABAergic and cholinergic neurons. For the astrocytic markers, we can see that there's a decrease of their expression already after 48 hours that was corroborated by immunocytochemistry that we could perform in samples exposed to amiodarone for 48 hours. And this is a staining for GFAP. This is the control, and you see that already at 1.5 micromolar of amiodarone, there's already a high decrease of GFAP staining. We also checked markers for oligodendrocytes, but we couldn't find a strong effect of amiodarone exposure. We also did some expression of markers linked to neuroinflammation, TNF-alpha, IL-1 beta, and IL-6. We find some changes, but we need to further investigate these changes.